Welcome everybody. This is a part of our ongoing cluster activity we have created for the benefit of Dr. Radha, that we have grouped our cancer experts in our institute, and they organize such activities. I mean, normally we would do physical, offline seminars. We would have invited you to be here in the campus, but nonetheless, this Zoom platform, and I was talking to Professor Murthy. Professor Murthy is here as a distinguished scientist. He looks after all research interest complexes, and he is the chief of all research. So his words are always very precious because he overlooks all over. So in that context, the cancer clusters are also in different campuses, and he knows the best, the experts and what they're doing. So I'll also request Professor Murthy. I mean, he has come with a long background of experience in the government sector, particularly DRDO, and he was the head, and he brings a lot of experience, and MIT is really benefiting with his 24 hours experience in our uplifting the research and our R&D push, which our founder president really wants to make it the best institutes, research institutes in the whole country, around the world also. Professor Murthy, please share a few thoughts, and then I hand over to Garge.
Thank you, Dr. Rajendra Prasad, and very good morning to each and every one of the viewers who have joined us today at this webinar on cancer-related domain. And I welcome particularly the speaker today, Professor Radha Ayagiri. And I have seen the profile. She has a very diverse the experience as well as the education. And I have seen her research encompassing from plant lectin, PhD, and right up to retinal degeneration, which she is going to talk about. Very versatile person who has accomplished tremendously in the field. Now she is speaking from Los Angeles, California. And she is also associated with Harvard Medical School. And so she is an expert in the field of ocular physiology, pathology, including the retinal degeneration. And Radhaji Amity is founded by Dr. Ashok Chauhan, the founder president, and also nurtured by chancellors Dr. Tul Chauhan, Dr. Asim Chauhan. And now we have 11 universities, including 2 campuses in US. We have one in San Francisco, and the other one is in the Long Islands of New York. So we have our presence in US, and we have developed a Amity Global Research Hub. So on behalf of founder president, Dr. Ashok Chauhan, and also Dr. Tul Chauhan and Dr. Asim Chauhan, the chancellors, I welcome each and every one of the participants who have joined us today. And Dr. Chauhan has created clusters. The cluster concept is, Amity has got since 11 universities, 17 campuses abroad. How do we connect these people, integrate them as one force? Because a cancer specialist may be in Haryana campus, another one may be in Noida campus, third one could be in US campus. So how do you integrate them? So we have developed this concept of clusters. So 15 clusters are operational now. And this will bring a very big force, because if you work alone, then you do little. But if you integrate yourself with all the capabilities, resources available as a group, then you can achieve many, many things and find solutions faster and a better solution. So with that concept, the clusters have been formed, and cancer cluster was the first one. So clusters are arranging a number of webinars, roundtable meetings, ultimately to bring out some very novel solutions to the problem of cancer. So that is the idea. So early diagnosis, treatment, therapeutics, and also the medical management. So the cluster is focusing on these areas. So your contribution today on retinal degeneration, which you have, and also the ocular pathology in which you have contributed immensely, is going to be extremely beneficial to all the participants. And we'll be also willing to connect with you once we have connected now. Thanks to Gargi, who has brought this Gargi Bakshi, who has been instrumental to bring this connect with you. So we like to have a sustainable connect with you so that we will be able to work together. We can contribute to your mission. Some of the synergy we can bring for your activities. So that is the whole philosophy of our the cluster concept. So I compliment all those from Amity University, Haryana. Dr. Rajendra Prasad is an outstanding scientist, which we are proud of in Amity. In Amity University, Haryana, he has brought a new paradigm in research and innovation. He has built many facilities, brought brilliant people, initiated a number of programs, and today Amity has 25,000 research publications. And many of them are in top journals like Nature Medicine and Cancer Letters. And we have papers in the Cell.
Lancet also.
Yeah, Lancet. Last year, 5 publications in Lancet. And so we have our H index is, our university H index is 87. So that shows there's a university we are doing very well. And then we have about 1,470 patents which have been filed. Many of them are getting translated into product technology processes, reaching out to the people. So we have focused on solutions, deliverables. So all our research tried to culminate either in the development of a product, technology, or a process. So that is how the benefit comes to the society at large. So we have sustainability and inclusivity as the forerunner in all our research. So whatever we do, it should be an accessible technology, affordable technology, green technology. So this is what our research in Amity is. So we'll be delighted to partner with you, and we are keenly looking forward to listen to all your achievements in your field. And with this, I welcome Professor Radha Ayagri and also all the participants who have joined us today, and have a wonderful day ahead. And Radhaji, now I'll hand it over to Rajendra Prasad and Gargi to conduct the whole proceedings.
So, Salamurthy, thank you very much for the very inspiring words and also letting us know about this MIT infrastructure and network of active researchers around the globe. Radhaji, this was for your information. We were a little bit aware of it, but I'm glad Professor Murthy has conveyed very nicely. We recently had this NYU, this Long Island campus. And that campus is we are trying to develop as a global research hub. Before I hand over to Gargi, I would like to tell Radha that in my one-time 2-year postdoc at NYU, I also worked with lens epithelial cells. And those days, the crystalline genes were very important in development of this cataract. So we used to dissect them or develop the primary cultures from bovine eyes. So it was very amazing and things like that.
Which lab were you in? Whose lab?
This was Joy Hochstedt in NYU Valhalla. It's a long time ago. I don't want to tell you when, so then you will know my age. So, Dr. Gargi, please introduce the speaker formally and we start the seminar.
Certainly. So, good morning, everyone. And of course, I thank Professor Radha, who took out time, especially because she has a very early start to the day. Tomorrow at 6 a.m. she has a meeting. She told me, and in spite of that, when I requested her, she was kind enough to agree and to share her experience and research work with us. And thank you, Murthy sir, for being here on the platform. And actually, I really look forward to the talks today because they are by people who have worked hard and explored the field so well. So I will simply move on to the webinars today. Let me introduce Professor Radha. So she started by doing her PhD from National Institute of Nutrition in Hyderabad, and then she went on to do postdoctoral research in USA. And I think initially she went to NIH and she did her training in ophthalmic genetics and clinical service branch at NIH. And after that, she moved to Michigan Ann Arbor Department of Ophthalmology as a research investigator, where she also became assistant professor and then associate professor. So her interaction interaction and association with Michigan Ann Arbor was for almost 10 years, after which in 2007 she moved on to University of California, San Diego, and there she was first associate professor, then professor, and now currently she is a chair of Department of Ophthalmology at UCSD, and she is also the director of Shiley Eye Institute Biobank, which I think is really important. And then she is a member, of course, of American Society of Human Genetics, Association for Research in Vision and Ophthalmology, and International Society of Eye Research. She is a reviewer of over 30 journals, and these include the likes of PNAS, Nature Genetics, Nature Communication. She has really done substantial, significant work in the area of eye and all the degeneration and intricacies that are involved. So I hand over to Professor Radha Ayagiri. We are really keen and eager to listen to your talk, ma'am. Kindly continue.
First of all, I'd like to thank Dr. Gargi for this invitation. So I started interacting with her very recently because I am trying to steal one of your students. So, I was so impressed with the training that particular student got. And so then I started talking to Dr. Gargi, and so that's how our relationship started. And hopefully we can continue further and with maybe more students and more projects. I also like to thank Dr. Murthy and Dr. Rajendra Prasad for your kind words. So it is really a pleasure for me to have this opportunity. Number one, actually, whether you realize or not, I came here to the US a long time ago, but I need to go home regularly to reconnect with my family in India, which we are not able to do it because of COVID. And so at least interacting with you all, makes me feel at home. And so it's a wonderful opportunity for me to be with you all and then share some of the research we have been doing. Of course, all of us are very excited about sharing our research at any time, but it's nothing like sharing with your own people. So thank you so much for this opportunity. And I'm also actually honored to share this platform with Dr. Elora Sen. I looked up her publications, and so she has an accomplished scientist. So it's wonderful to have this opportunity to, share with her. So I, yes, when Dr. Bagchi asked me to talk, I was a little hesitant because, I have an international meeting with Japanese colleagues and European colleagues, and the only time that works for all of us is 6 a.m. But at the same time, as I said, this is a joy for me to join this, and thanks to Zoom, at least we can interact. So I am going to start sharing my screen and then see how well I do. If there is any issue, we will figure it out. So today I thought I would talk about the genetic landscape and pathology of inherited retinal degenerations. So basically, if you think about my lab has two different components. One of the components is we study genetics of retinal diseases. So the goal is to find what is the underlying cause of these diseases. And the second component is once we find the underlying cause, so we go further to understand how these mutations, individual mutations, leading to retinal degeneration. So we study the mechanistic aspect of this to study these underlying molecular networks. So we use different model systems, either mouse models, cell models, or patient iPSC differentiated uh, tissue models. And more recently, we are beginning to make 3D retinal structures. So I will try to talk about the genetic landscape, and if I get time, I will also go into the mechanistic aspect of this. So let us see. So as I said, the focus of my research is the eye, human eye. Um, Here what I'm showing is, this is something, most of you are all familiar with it. So the tissue I'm interested in is retina. Retina is at the back of the eye. It is in the posterior half of the eye. Here is the retina. When light falls through the lens onto the retina, so here the signal gets processed, and then here is the optic nerve. It communicates the information through the optic nerve to the brain. So this is the cross section of the retina. So the top layer of the retina is a single layer of retinal epithelial cells. So these are the very important cells because as they provide nutrients to rest of the layers of the retina, and also these the metabolic products of these different cells, they are recycled back to the circulation or vasculature through the RPE cells, these epithelial cells. So they provide nutrients to rest of the retinal cells, and they take back they cycle back the metabolites. And then below that is the rods and cones. So these are the cells that capture the light signal and gets processed by different types of cells in other layers, and the signal will be communicated by processing the image through these different networks of cells, and eventually the axons of the ganglion cells in the last layer, they bundle together and they form the optic nerve, and then that's how through the optic nerve the image is communicated to the brain. So the cells that are the genes that are expressed in all these different cells, mutations in any of those genes can lead to retinal disease. So here today I will be talking mainly about two different conditions. So first, this is the image of the retina. So when you are looking through the lens, this is how the back of the retina looks. So you might have seen when you go to the ophthalmologist, he takes a handheld lens-like instrument, which is called fundoscope. So that helps in focusing on the back of the eye. And so this is how the retina looks when the ophthalmologist is looking through the lens using the fundoscope. So here, this is the optic nerve, and the central part of the retina is called macula. Macula is rich in cones, and we use our macula to view fine vision. This is what it provides. Like fine vision is like something like reading something, or looking at details, or putting a thread through the needle hole. So the rest of the retina, this is the peripheral retina, which is rich in rods, and this is for the peripheral vision. A normal individual with normal retina, so this is how this figure will appear. In case of macular degeneration, this is the fundus from a patient with macular degeneration. You can see the macula has degenerated, and so this patient lost central vision. So it results in a blind spot right in the middle of the visual field. And as he moves his eyes, so the blind spot stays right in the middle. Here, this particular image is from a patient with retinitis pigmentosa. So you can see the peripheral retina has degenerated. This is somewhat opposite of macular degeneration. So here the central vision is preserved. So this patient can actually read a newspaper, but will not be able to move efficiently because the peripheral vision is impacted, and he ends up with tunnel vision, and while moving, like, he would not see the objects in his way. But in some cases, unfortunately, they even lose the central vision eventually. So today the diseases, when I'm talking about retinal degenerations, I'm referring both macular degeneration and retinitis pigmentosa. So these retinal degenerations, there are a wide variety of them. So they exist in different modes, like the dominant, recessive, X-linked, mitochondrial, and complex, different patterns of inheritance. So there is a lot of heterogeneity, like these diseases vary, widely. And there are also a lot of different genes that are associated with this genetic heterogeneity. And there is variable penetrance. In some cases, like, even when they have a mutation, they do not develop disease. And variable expression, that is the expression of the disease, like the severity of disease may vary within the family, like different patients may have the same mutation, but they may have different expression profile. So and some of these diseases, there is an overlap in clinical symptoms. So what happens is when these patients come to the clinic, it becomes highly challenging to provide a diagnosis just simply based on clinical diagnosis, because some of the diseases, like, they progress very fast, some of them progress slow. So you cannot give a prognosis to these patients. So diagnosis is important, of course, like, even also for prognosis, and to provide any information to these patients that is useful, in planning. But these degenerations are irreversible, and eventually they lead to complete vision loss. So there are about 377 loci for these genes, for these retinal diseases have been mapped. And out of them, 271 genes have already been described. So now when we analyze these patients, so in about 70% of cases almost, like, we can find mutations in genes in these 270 genes. So which is really a major advantage, because, like, we can provide them, the specific diagnosis based on these mutations. However, like at this point, my research focus is on the remaining 30% of cases where we are finding it challenging to find these mutations and understand the disease process. So as I said, like, I will be talking, I'm hoping to cover two different aspects here. One is understanding the genetic landscape of these diseases. And so we call it elusive genes project, because this is to find genes that we are not challenging to find. So the missing causality in patients. And the second one is to study the mechanistic understanding of these disease processes, using one of them as an example. So here, like, we have utilized the whole genome sequence approach, because that allows us to examine the entire genome, not the selected parts of the genome. So that way we can have a comprehensive analysis of the entire genome to find the mutations. We studied 3 different populations. So one of them is a population from Mexico, Yucatan Peninsula. So this is an understudied population. And Mexican population, as you know, is an admixture of native population plus European individuals of European ancestry. So it is an admixture population. So the other population is Pakistani population from the Punjab region of Pakistan. So here, this is again another unique population, because this is a Pakistani population is endogamous population. So highly inbred, somewhat, opposite of the Yucatan Peninsula, and then still understudied. And the third population is a Caucasian population from the US, which is well-studied population. So these are the 3 different populations we analyzed using whole genome analysis. So we used Illumina sequencing, and then we have used different standard tools that are available to map the variants to the reference genome and then call variants. So I'm not going to go into the details of this in the interest of time, but in case if anybody is interested, we'll be happy to share. And this also has already been published. So we used some of the publicly available tools in addition to tools we put together. So these are to detect large deletions in the genome and variants in the regulatory region, and predicting the impact of changes we find in the genome, and if a change results in altering the splicing. And we studied X and Y chromosomes independently while autosomes were used as pairs. So when we analyze a patient, so we generally find about 4 to 4.5 million changes in an individual. But now our goal is to find that single variant in that patient if it is a dominant disease, or two variants, like if it is a recessive compound heterozygous mutations that are responsible for disease in that. So now we have to find that one or two mutations in that patient to find the cause of the disease. So that is like finding a needle in the haystack from this 4.5 million variants, finding that one or two variants. So that is where we have to have a very good strategy and analysis pipeline to filter out the variants that are not involved in causing disease, and to focus on what are the likely causative mutations and establish what's causing disease in a patient. So our hypothesis is like, either, like as I said, like we already analyzed them once. So in a majority of these patients, and 70% of cases, mutations were found using other methodologies. And we are trying to find the remaining 30% of cases. So in that case, we have to, the hypothesis is the mutations are in novel genes or some mutations in the non-coding region, or challenging to find mutations in many cases. So I'll give a couple of examples. So here, this is an individual. So she is affected with retinal degeneration. So she has no family history of retinal disease. So if there is no family history, if parents are not affected, generally, it could be a recessive disease. That's the first guess. But since we have no information here, we also have to think it could be a dominant because of a de novo mutation. That's a new mutation in this particular case. And so, and that's why she has the disease and no other family members have it. Or she has autosomal dominant mutation, but variable penetrance, and one of the parents have the variant, but they didn't develop the disease and she did. Or it could be X-linked. And again, similarly, the mom could be a carrier but did not develop disease. And in some of the retinal diseases, some of the carriers develop disease because it depends on the X inactivation, the pattern of X inactivation. And it could be different between these two individuals, although they both are carriers. One could develop disease and other. So these are some of the known facts about retinal degeneration. So we have to consider all these patterns of inheritance when we are looking at we are trying to find the causative mutation in this individual from the whole genome variants. So this particular individual was diagnosed with retinal degeneration at age 13, and when she came to us, she was 38, and she was almost losing majority of her vision. So here, this is so what we have done is we have done whole genome sequence of this individual and her sibling. So here, using the sibling will allow us to filter out a majority of variants, because if he has that variant, and since he does not have the disease, probably that's not causing disease, causing this pathology. So hence we can filter it out based on the pattern of inheritance we are modeling it for. So using that strategy, so we have designed a filtering strategy, and which I will show. So we, oh, sorry, I'm not in a presentation mode. So here what happened is, like, we have collaborated with one of my colleagues, Kelly Fraser, on this computational analysis. So in this individual, we found about 4 million variants. Out of this 4 million variants, we found about, after filtering using her sibling and additional strategies, we came up with about 9,000 variants we could consider as potentially disease causing. And out of them, like, about 500 are some rare, because this disease is rare, they have to be, rare alleles. And for about 172 them are potentially pathogenic. And out of them, 8 of them were detected in retinal degenerative genes. And an additional variant, like structural variant, was found in a gene, one of these 8 genes, a second variant was found. So using this, like, these 2 variants were detected in a gene called MERTK. One of them is a splice variant and other one is a deletion variant. Sorry, like, actually most of the slides I have hidden are also showing up because I'm using a different presentation mode, but that's okay. Here, this is the splice variant, and this variant could result in a skipping of the exon. And here there is a deletion variant. So this deletion results in loss of exon 9. So once again, the loss of this complete exon also could result in alteration of the transcript. So when we tested the segregation of these 2 variants, so we could see, like, the splice variant is inherited from the mom and the deletion variant is inherited from the dad. So all the genetic studies today I'll be talking were carried out by my graduate student Pooja Biswas. So she performed all these analyses. So here both these variants are segregating with the disease. So hence that is likely they may be the underlying cause of disease in this particular pedigree. That is the assumption at this point. But we need to make sure what these loss of these or these mutations could lead to pathology. What is the likely impact of mutations in this particular gene? So one is both these variants could result in generation of a shorter but functional protein, or they may result in the complete loss of this protein. So as I said, this particular gene is expressed in the RPE, and RPE function is critical for the function of the rest of the retina. So, but if we want to study whether these mutations have any impact, we need to get the RPE tissue of these patients. But as such, this patient already has retinal degeneration, and so we cannot take a biopsy. So which will only further cause further loss of vision, and so it's not a feasible idea at this point. Like, how do we study what is happening in the RPE of this patient? So one of the ways to do that is using the iPSC or the stem cells from this particular patient and differentiate them to RPE tissue to study that. So we collected the blood samples of these patients, and both the patient and her father and mother, and then we reprogrammed them to iPSC for our further studies. So we generated iPSC and then we characterized them primarily using the different standard iPSC markers. And so these iPSC, we have differentiated them to retinal pigment epithelial cells. So those are the cells which are the closest model we can get to study the RPE of these patients. So here what I'm showing is these are the RPE that are differentiated from this patient and control iPSCs. And RPE have specific structures, hexagonal pattern, and these are the different markers. So of RPE characteristic markers, which we stain them with. So here, to study RPE, we need to have, polarized RPE cells. So for that, we have generated these cells and then again used them with markers. And then these markers show, we can see that the different polarization and polarized RPE cells now we could generate based on the marker profiles. We have then these polarized RPE cells were stained with MERTK antibodies. As you can see, this is the apical and basal side, and then we should see that these are the control line from a normal individual iPSC differentiated to RPE. You can see the presence of MERTK, which is shown in red, and green is a marker protein, which is a ZO1. So in case of the patient iPSC differentiated to RPE, we did not find our MERTK signal. So this suggests either absence of MERTK in these patients' RPE, or our antibodies are not detecting if there is a truncated protein, because you remember, it has altered the transcript and it may truncate the protein, but still we may have a protein which is not detected by these, by the antibodies we used. So what we did is we have done again, sometimes immunohistochemistry, some antibodies work for immunohistochemistry and some do not work very well. So hence we have done Western blot analysis of the father, mother, and the patient, both in iPSC line and also iPSC differentiated to RPE stained with MERTK. So we can see the presence of MERTK signal, but not in the proband, both in patient, father and mother, and also the control line, both in reducing and non-reducing conditions. So similarly, when we did the iPSC differentiated RPE, we did not find a signal for MERTK in the patient. So this suggests either there is a loss of MERTK or the antibodies did not detect this MERTK. So to test that whether there is a transcript, because that will tell us whether there is a protein or not in an indirect way. So hence we measure the transcript levels of this MERTK. And you can see in case of the patient, the transcript levels are very low compared to, significantly low compared to the father and mother and also the control line. So suggesting that in these patients, the MERTK, levels of MERTK are very low to minimal levels. And that may result in loss of MERTK. So what is the impact of loss of MERTK physiologically? One of the physiological tests to measure the function of RPE or physiological integrity of RPE is measuring the transepithelial resistance. So we measure transepithelial resistance in these iPSC differentiated RPE, and then so we do see that they have normal range TER. So suggesting that their TER is physiologically, they appear to be normal at this point. So one of the major functions of RPE is phagocytosis. So we studied the phagocytosis in these RPE. For that, what we have done is we have a iPSC differentiated RPE were fed with fluorescent labeled photoreceptor outer segments. So when the control line was fed with the fluorescent labeled photoreceptor outer segments, you can see the phagocytosed fluorescent signal compared to in case of the mutant line or the patient iPSC RPE. So they did not show active phagocytosis. So this suggests like, the phagocytosis is impacted in these cells. So this could be because our MERTK gene is known to be a key player in RPE phagocytosis. And in our case, in this particular patient, there is a loss of MERTK because of the mutations we detected. And that is leading to abnormal phagocytosis. And abnormal phagocytosis would result in, is known to result in RPE degeneration. And if RPE is degenerated, because that results in degeneration of retina, and that could be the mechanism involved in a pathology observed in this particular patient. So using this kind of model, like, we can establish the causative, find the causative mutation, and then study the impact of the mutation. Because the whole goal here is to think about how to prevent these diseases or how to help this patient. So by knowing the underlying cause of these pathologies. So now we could determine the the cause of pathology in this particular patient. I'm going to skip this. Like, as I said, I'm sorry, I have hidden these slides, but since we are using a different mode, now they show up. So here, this is another pedigree we studied, where again, there is only one affected individual. And when we sequenced the whole genome, we found a mutation, which is a stop codon mutation, stop gain, in a gene called EYS. So mutations in EYS result in recessive retinal degeneration. But one mutation is not sufficient. We need to find the second mutation. So we analyzed this gene up and down because we have already sequenced the whole genome, but we could not find the second mutation. However, we found two different microdeletions in the upstream region of this EYS gene, one 5 base pair deletion, and another 4 base pair deletion in the intron 12 of this gene. So these are non-coding changes, and it is highly challenging to interpret, like, what is the impact of these variants. So, but in any case, first we need to establish, like, these mutations indeed occur because it was found by the whole genome sequence analysis. So what we did is we tried to design primers on both sides of this and then amplify and then sequence, so that way we can establish, like, the presence of these. This is a standard protocol and anybody can do it, but however, we failed to do that. We could not amplify by designing primers to the both sides of this. So that intrigued us, but at the same time, like, we could not find it. So then we collaborated with Illumina, which is down the block from my department. So they analyzed Illumina, they had a new software, and they could not find it. So then we went to another company, BioNanogenomics, who developed a new tool. Some of you may be familiar with it. We used to use something called fiberfish, where you stretch the chromosomes and then stain them to see the chromosome structures, to know the larger structural patterns in chromosomes. So based on that, so they generated a very fine methodology to have detailed analysis of the genomic patterns. And so we utilized that technology. And then what that showed us, there is actually, these two deletions are there, but at the same time, what happened is there is a large inversion, balanced inversion. So when you are doing whole genome sequence analysis, when you are mapping the reads to the reference genome, it only sees, is it present or not. If the sequence is present, it will go and then show that sequence is present. But it won't tell us the orientation of the sequence. Hence, using our both Illumina technology and our own technology, we could not find this large inversion. If there is a small ones, we can detect. But when it becomes too large, we cannot detect them. So this, the technology used with BioNanogenomics, that allowed us to identify this. So at that point, we could amplify. Now, when you design primers on both sides of this in that orientation, because it is in the reverse orientation, we could amplify and then sequence. So now we know this particular inversion is there. So what does this inversion do? So this inversion now from exon 1 to 12 of this EYS is inverted. So obviously, that impacts the transcript of EYS and then results in loss of EYS transcript. Now we found the second mutation. So EYS first mutation is the stop codon mutation, and then now the second mutation, this inversion, both of them result in loss of EYS, and then that results in retinal degeneration in this particular patient. So this is really significant because right now we are conducting a clinical trial for EYS gene therapy, and then so this patient can be benefited using that. So as I said, these are some of those otherwise cannot be found easily. So we need to utilize different technologies to detect these mutations to help every single patient. It has to be somewhat personalized. So, okay, I'm going to skip a lot of slides here. I'm sorry about that. As I said, I meant to hide them, but now we are using a different format. So what are the types of common mutations we find is coding sequence mutations, de novo variants, and deletions. These are all common, and then we can find them. That's not a major challenge. But the mutations in the non-coding region, that is the dark side of the genome. So we do not know, actually, first thing is only 1.5% of the genome is coding sequence. So if there are mutations, mostly we can interpret the impact of those mutations, as I have shown in case of MERTK, using various methodologies. So at least we can attempt to understand. But in case of the non-coding genome, there is not much information available about majority of the non-coding sequence. So we used to think at some point, that was all junk DNA. But now we need the most of the mutations, as I said, in 30% of the cases, we are not finding mutations. And those mutations are likely to be in this non-coding region. And then that is what we need to learn more about at this point. I'll just give a brief example. So these non-coding mutations can be in intronic changes, like exon skipping, splice gain, poison exon, creating poison exons. So those are some of the standard ones and easier to find. But mutations that are present in regulatory regions that are still challenging. So I'll just give a brief example. So there are thousands of enhancer regulatory elements in the genome, and that regulate the expression of every single gene. So here what I'm showing is in the schematic, there are 3 different genes, and then there are multiple regulatory elements. Some of them can be close, some of them can be far. So these regulatory elements impact control the expression of this. So here what I'm showing is these are called topologically associated domains, or TADs. So here there is one gene A, and then so you have these 2 different domains. So both of them can control the expression of this particular one. So in this particular case, in this TAD, there are 3 different regulatory domains, and they can regulate this in different ways, this particular gene. I'm going to go back to the previous slide. So here, each one of these may involve in the regulation of expression of this gene in different parts or different tissue. So hence, again, these stats are cell and tissue type specific, they can be. So here, this schematic shows this is the regulatory region. Then these are the different enhancer elements which are regulate the expression in one particular specific tissue. In case of here, the spinal cord, brain, and other tissue, so on and so forth. So each one of them, they regulate the expression of this gene, these different enhancers, and then all these TADs together, like they control the expression. And mutations in any one of them, like not any one of them, but if it is in the brain, for example, if there is a mutation here, that's not allowing it, or that is impacting the enhancer function of this particular enhancer, then this can impact the function of that particular organ or tissue where it is regulating the expression. So from that point of view, we are trying to understand the retinal, retina has, as I said, several different types of cells. And then among these cells, like, there are 6 major types, and then there are several subtypes, and there are about 125 million cells in the human retina. And these cells, each one type, they have their own type of TADs, they have their own type of regulatory mechanism. And we have not understood the regulatory profile and the 3D genome of retinal cell types. So right now, my lab is focused on finding these, mapping the 3D genome of these individual cell types in the retina, because this information does not exist at this point. So we actually we have initiated these studies, and then so we have done RNA-seq, and then these are the different clusters. This is single cell RNA-seq, so that we can get the transcriptome profile of each individual type of cells here. And you can see these clusters separating the cell types. And we have also performed ATAC-seq. So once again, to understand the regulatory profile, and then we are trying to generate the epitranscriptome maps, and we are still yet to complete the Hi-C. So that will allow us to understand the interaction between these different regulatory elements and the genes, and in each different cell type, that will allow us to map the 3D genome of these individual cells. So then now we already have the whole genome sequence of these hundreds of patients. So, and using the mutations in those patients, so we can interpret the changes that we are finding and how those non-coding changes in these potential regulatory regions are contributing to retinal degeneration. So here I will give an example of that with a particular complex disease condition. Here, we are talking about age-related macular degeneration. Age-related macular degeneration is a common condition that actually leads to impacts the vision in more than 20% of population in the over the age of 65 in the US and actually worldwide. So there are about 52 AMD2 risk variants that have been identified in about 34 loci. So what we have done is we have taken iPSC lines, and then we have the whole genome sequence, ATAC-seq and RNA-seq of these, about 200 iPSC lines. So from these, we used this, and then we performed fine mapping and identified for this particular study, one risk variant I'm talking about. So there is a one risk allele we identified for AMD. So this risk allele, we have then now that we have we have differentiated them to RPE because this particular gene is expressed in the RPE, and we performed ATAC and RNA-seq. So here what I'm showing is, I do not know whether you can see well because the slides are not big enough, but here this is where the SNP is, which is associated with AMD. This is the AMD risk variant. You can see that it is in the region where there is an ATAC peak. And here also it is at the upstream sequence of this is the RNA-seq profile where the expression, these are all the different exons. And you can see it is in the upstream region of the gene in the regulatory region. So when we have used these and we studied the attack profile, and what you are seeing in gray is the risk allele, and this is the wild type allele. You can see that higher attack depth. And when it comes, we also studied the expression of this gene. So here, this is the patient cell line, iPSC differentiated RPE with AMD. So with the risk allele, you can see very low expression of VEGF gene. So alteration in VEGF gene, VEGF expression is associated with age-related macular degeneration. And interestingly, so there is the treatment for AMD is associated with modulation of VEGF levels. So that is right now standard treatment for AMD. So we can, although these are all non-coding variants, so we can interpret by identifying these, the regulatory profiles of these and how it is regulating, and then one could design, think about therapeutic strategies using this particular information. Now I'll just give you a kind of a summary of the ethnographic distribution. As I said, we studied different populations to understand the ethnographic distribution, because this kind of information will again allow us to interpret the data in different populations and extend it to other populations based on these population structures. So when we studied the Mexican population, so we could find novel mutations in about 50% of cases. We found mutations in 68% of these overall cases. So, but among them, 75% of the mutations we found are not listed in NOMAD. NOMAD is the database of variants in the whole world populations, different populations. So this is the best representation of the world population distribution of mutations or any changes in the genome. And 75% of them are not listed. But it's not surprising because, as I said, this population is one, understudied. Two, there is a good proportion of native ancestry contributing to this population along with Europeans. So because of that, this is understudied, and hence this is not well represented in this population in the public domain databases. And so that is consistent with what is known already. But at this point, if we want to treat these patients, so we need to think about, enhancing the information on this particular population. So that way, they can be treatments targeting these novel variants can be developed. So coming to Pakistani population, about 80% of variants are novel, because that's not, because the Pakistani population was not studied for inherited retinal dystrophies. But however, one thing is Pakistani population is very well studied for several other diseases. So because of that, a majority of Pakistani variants are in public domain data, and that population is well studied and well represented in NOMAD. Hence, compared to Mexican population, here 75% are novel, but in case of Pakistani population, only 14% are novel. Also, majority of variants found in Pakistani population, that is 86%, they are specific to South Asians. So the advantage of this is whatever we found or anybody is finding in this population, that can be applicable to the entire South Asian population. So there are some of the populations well studied, some are not studied, but at the same time, using the small population, we can utilize that information for larger population in the entire, most of the South Asian population. So coming to Caucasian population, we found 50% of novel variants, which was surprising because Pakistani population is understudied and Caucasian population is well studied. However, here we found 50% novel, while and 86% of them are in the public domain, but here only 50%. So this is because the pedigrees that came to my lab, that is, it is almost like it's a tertiary analysis, because people have already, these pedigrees were well studied and analyzed by multiple investigators when they did not find mutations. So eventually, like, they passed them on to my lab. So hence these mutations are not common mutations, and these are very rare, and also probably like, more recent mutations. So that's why they are not very common in the general European population. They are private mutations. So hence we found them, and they are not in the public domain. But all this allows us to think about, one, diagnostics, designing easier diagnostics, and interpreting data, and then eventually determining, like, therapeutics, which therapy is appropriate for what population, and how to design these strategies. Here, what this is a little bit of a different story, like where this is a pedigree where we found a mutation, a deletion mutation in this family with 4 affected individuals, and the gene is called MFRP, and this particular mutation leads to retinal degeneration and microphthalmia. So what we have done is, like, we introduced this mutation using CRISPR-Cas9 into a mouse model. And so these mice here, what I'm showing is fundus autofluorescent. And as you see, the fundus autofluorescent spots have increased with age in this particular mouse when we have introduced the mutation compared to the wild type. Wild type, there are not much changes. And here, what I'm showing is a retinal sections of these mice. So in wild type mice, like here, I want you to focus on this particular layer. There are 10 different rows of nuclei. But in the mutant model, like, at 21 days, there are about 9 to 10 rows. But then by 2 months, there are about 5 rows. And by 3 months and 12 months, there are only 1 row of nuclei left, because there is a significant degeneration of retina because of the mutation we introduced. So we developed a AAV-based therapy to treat this disease. So hence we have injected the AAV vector. So one of the advantages of treating retinal degeneration is retina is very accessible because it's eye, and one can inject the whatever you want to deliver into the eye, and eye has immune privilege. So hence, because of the blood retina barrier, whatever you inject into the eye, majority of them, they do not have any systemic effects. So we administered gene therapy to this model, and you can see, and then after 4 months, we stained it for MFRP expression, because the mutation we introduced results in loss of MFRP. And so here, this is the sham treated, and then you do not see any expression of MFRP. But in case of AAV treated, you can see the nice expression of MFRP after 4 months in this mouse. And we still see at 9 months, still good expression of MFRP. And also here, what we stained it with cone opsins. And then, so M-opsin, which represents cones. So it also preserved a significant number of cones. And then you can see, like, there are 6 to 8 rows of photoreceptor nuclei preserved. So that, even after a single injection, after 9 months, we can see, like, good preservation of retinal structure, retinal function, like, which I'm not showing here, with this particular therapy, and one could think about it. And then, so right now, like, it's this is ready to move to the human phase. So there are, for retinal diseases, there is a gene therapy already available for one of them, RPE6. If RPE65 is a gene, mutations in this gene leads to severe retinal degeneration. And there is an FDA-approved gene therapy available for this. And there are 20 other therapies that are in progress. So there are also some stem cell therapies. We are actually transplanting stem cells into mouse models at this point. which I'm not going to talk about in the interest of time. And then again, similarly, gene editing, that's very easy to deliver gene editing and into the eye. So that's again a very common one which is being tested. And there is a human clinical trial right now underway in my own institute. And there are also neuroprotective drugs that can be delivered to preserve the retina. So there are different treatment modalities that are available, which of course they're all based on the genetic mutations which we detect. So it is very important to find the underlying cause of these diseases. Basically, like, we want to find this mutations in every single patient to be able to provide diagnosis, and eventually, somebody should develop the treatments based on these mutations. I'll just give you a glimpse of this because this is a big story, but I'm not going to go into details. So here, actually, what we studied is we studied a pedigree, large pedigree with the retinal degeneration, and we found a mutation in one of the in a gene called CTRP5, and then we made a mouse model with this particular mutation, and these mice develop disease very similar to what we see in patients. Here they develop in the below the RPE, they develop deposits, so which will result in abnormal RPE. So, and also like this gene, the mutant gene, this is the mutant protein that significantly accumulates in the RPE region, and then so it forms it is in the extracellular matrix. We carried out several studies. Of course, we performed the proteome analysis of this mice. We performed Is2 hybrid analysis to identify what this gene interacts with. Eventually, by using multiple studies, all of them, we also generated iPSC of these patients' stem cell based RPE, and this patient stem cell RPE model also shows this development of these deposits in the RPE, in this stem cell differentiated RPE. And we studied the interaction of this gene with other what are the other genes it interacts with using Is2 hybrid, and then we established like this gene interacts with another gene called HTRA1, and it also it interacts with HTRA1, it activates with HTRA1, and HTRA1 cleaves CTRP5, so they act on each other. So the interesting aspect of all this is eventually when we have seen that, like, this is the mouse model, we see this is the wild type and this is the mutant, this is the wild type and these are the mutant mice. So both CTRP5 and HTRA1, they both accumulate in the same region. So the bottom line here is what we found is there are different substrates of HTRA1, including the gene we found CTRP5. All of them are substrates of this HTRA1, and all of them result in deposit forming phenotype. Why is this important? This is important because AMD is the leading cause of blindness, and the phenotype there is formation of deposits. To understand that, this is the model that will help us to understand how these deposits are formed. Now we have a network of genes that all lead to the same phenotype, but all of them interact with HTRA1. So that is what we identified using different methodologies in my lab, and different models, iPSC and mouse models. So now this is another project we are focusing on to understand how this particular network of proteins leading to pathology through their interaction, and these are all extracellular matrix proteins. So I am going to, sorry I rushed here, but so I like to acknowledge the contribution of my lab. Pooja Biswas is the graduate student who contributed for most of the studies. Anil Chekuri, I did not go into details, but he performed majority of functional studies, and Anil is right now at Harvard as a associate, junior investigator. Shyamanga Barua he was a postdoc in my lab, and right now he's an assistant professor in my department, and right now he's my colleague. And Murthy Chavali, he generated the mouse models, and he was also my previous postdoc, but right now he's an assistant professor at UPenn. And I collaborated with Dr. Duncan and Paul Saving, who is my mentor at the University of Michigan, and Hakan Lively with patients. And I collaborated with Ada Villanueva in Mexico on Mexican pedigrees. Amir Riazuddin, he's my collaborator on Pakistani pedigrees, and he's an associate professor at Johns Hopkins. And Amir and we were trained together in Dr. Hedmanchak's lab at the National Eye Institute. And then we three collaborate, for the past more than 25 years. So that's how my Pakistani collaboration is strengthened through my mentor and, of course, my colleague, Amir. And we collaborated with Astra on generation of the AAV vectors and funding for, IRD genetics. And then, of course, the late onset retinal degeneration model, which I did not talk more about, the functional studies using single cell genomics. And then the third funding from NIH is to study the human 3D retinal genome maps. And then elusive causality of disease is funded by the Foundation Fighting Blindness. And I really like to thank you all very much for letting me share our work with you.
Professor Radhaji, it's a very, very extensive studies you have carried out. I wish you had more time to discuss a lot of voluminous data you have collected, both in this macular degeneration, retinal degeneration. And now let me start the discussion. What are your therapeutic approaches based on these molecular studies which you have carried out? What are the efforts in your group in terms of the therapy, translating these molecular biological studies and genetic studies to translate them into an application for the treatment?
So what are the strategies we are using, you're asking, right?
Yeah, therapeutic strategies.
Yeah, as you mentioned, one, I have shown the development of AAV-based therapies. That is one of the primary methodology we have been trying. And then the other one is, of course, gene editing, because that is something a little bit more precise. And and the more recently, we started utilizing, antisense oligotherapies. And here we have an initiative on precision medicine.
Right.
So for every individual, because it's a different one, so hence there are organizations who are interested in precision medicine for personalizing these therapies. So the other one, the more recent, very recent one, is allele specific oligo. And we also have, as I said, Shyamanga Barua, who was a postdoc and my colleague right now. So in collaboration with him, we are testing some of the stem cell transplantation studies. So we test them all first in iPSC models. Like, for example, if it is gene editing, we test it, or allele specific or antisense oligonucleotide, we test them on iPSC model, then the mouse model. And then it is up to the bigger community to take it to the next level for evaluating therapies in patients.
I think, Gargi, you may like to take questions from.
Yes, certainly there are a few questions. We will take at least a couple of them. So first of all, thank you, Professor Radha. It was such a vast and intricate, and I would say it was a new area, sort of the details were new for me. So thank you so much for this talk, and the future scope is enormous with this. Since we are a bit short of time, I would like to take up one or two questions which have been asked, but I hope we can refer to you again and again in the future when people have more questions in their mind. So one of the questions is from Vimal Singh, and he is asking whether biopsy samples were taken for genome analysis, were they from blood or other extractable tissue?
Gargi, if I can
Yes.
interrupt for a second.
Yes.
I mean, she will have many questions. But
Of
I'm
course.
just trying to say that Professor Das, BC Das is also there waiting. So maybe if he has some comments to say,
Okay.
let me ask him. Otherwise, we go ahead with the questions.
Okay.
Professor Das, are you here?
Yeah, good morning to everybody.
Our Radha's talk is just over. So,
Yeah, I was busy with the other things, but I was listening to the talk. But I am sorry, I have not made, in between there are some other things, so I could not keep the balance. Means I have somewhere I have lost the clue also, because you have to listen to a talk continuously, and it's,
yes,
but
yes.
certainly I would appreciate Dr. Radha that extremely the huge data she has accumulated, that not only the molecular biology clinical part also, but along with that, that the from the stem cell to iPSC and to exosomes, I think there are all modern aspects have been considered. But question is, some what I was wondering, that after creating the iPSCs, is there, what is it exactly you want to do with the iPSCs, and what the clinical applications of that? If you have anything you have that you orient towards the clinical applications. So that I could not get much of the things, but as far as the basic study is concerned, they are excellent. So if you could give some glimpse of that what clinical applications would be from the point of view of the, because many stem cell applications in the retinal biology or the eye research has been being used, and there are many, you might, in the Indian situation also, like the LB Prasad Institutes and then Shankara Netrala, they have a lot of, particularly in the LB Prasad Institute in Hyderabad, they do a huge number of things which has of the more of the clinical applications. So could you give some impressions of that? That would be nice.
I will try, Dr. Das. So actually very nice meeting you online. I'm very closely associated with all three institutes, both Shankara and LVP and Madurai. We interact with them very closely, and I visited them several times. I'm somewhat aware of their work, probably not completely, but so when it comes to the retina, as you know, like we have the 3D structure, and from the iPSC, we can differentiate them to different cell types. And people have, not my lab, but people have done, like, we can differentiate them to retinal progenitor cells. Actually, I did have slides which I did not show. Differentiate them to retinal progenitor cells, and then when you inject them, they are getting integrated really well into the retina. So one can, if depending on the type of mutations, because some mutations impact photoreceptors, so we can generate photoreceptor progenitor cells by correcting the mutation. For example, in a patient,
you have done anything with the genome editing CRISPR-Cas? We used any gene mutation, or anybody is doing that in your group?
Yes. Actually, of course, no. In my group, we have done only RPE because that's the easier one. We have injected RPE into the mouse model. Actually, I was talking about toward the end. So we are still waiting for results. but the whole goal is eventually, like, we have patient iPSC and then correct them using, gene editing.
Yeah, right.
And the corrected cells, you can inject them into the RPE or differentiate them to RPE and then
Right,
inject.
right.
That's the whole goal.
Yes.
And then coming to other cells, like photoreceptors, also people have shown this. Like, it's not that actually they are getting ready for human clinical trials. In Japan, they have injected RPE into patients. But unfortunately, like one particular patient, there were adverse events about 4 or 5 years ago. This is in the Yamanaka group did that. And then so they have suspended it. And you probably know, like now the whole Japanese genome has been characterized with reference to the immune reaction with reference to HLA. And they came up with it and they figured out a way to avoid any adverse events when they are, injecting these.
Injecting, yes.
So hopefully it will work because Japanese population is more structured, small population, unlike US population, which is, very, heterogeneous. But I think once we, this is very much feasible at this point, but however, we need to understand further to avoid any adverse reactions. But right now, the clinical trials have already been approved at NIH and then for AMD, and that is going to happen.
Including the stem cell injection, the stem cell therapy?
That is a replacement that is into the retina.
Yes. Yeah. But it's unfortunate it is in India still not approved by the ICMR and not for the retinal but other applications of the stem cells, because we have a stem cell institute here and we mostly work on the cancer, but the relation to cancer we have 3D printing arrangements and all the organoid development. iPSC is regularly being developed and also the CRISPR-Cas system is used, but it is mostly relation to the cancer. So that we are at MIT Noida, so that we do, but it's excellent to listen to your talk. So I'm sorry, in between I have to make some other things, but certainly I can understand your the immense contributions in this field, and it's great, nice. Thank you so much that we could learn many things. Thank you, and it is good for all the young people and the students, PhD students and others. It's excellent. Thank you so much.
Thank you for your kind words. Like it's a group effort, and it's.
Thank you.
I can just take 1 or 2 questions, and then because we are also running out of time.
Yeah. So as I
thank for her also,
Professor Das, and once again I will take up Dr. Vimal Singh's question whether biopsy samples for genome analysis were from blood or other extractable tissue. He's asking.
So for actually for whole genome analysis, we used blood samples because it doesn't matter. We are using the genome. But when we are doing other transcriptome analysis or epigenome analysis, for those we need, because retina has its own different regulators and different expression profile. So at that point, we are using iPSC differentiated retinal tissue, retinal cells, or if it is a mouse model, we are using mouse retinal cells. It is not possible to take a biopsy sample because the retina is very delicate and already these patients have a degeneration. So hence we cannot use a biopsy sample, retinal biopsy samples. Hope that answers the question.
Right. Radha, one thing I wanted to ask you was that you mentioned that many of the mutations are in the non-coding regions of the genes also. So the mutations in the non-coding regions, do they always have the same manifestations in the patients? Or they...
Yeah, yes and no.
May manifest differently.
If the non-coding means, if the mutation is same, right? So theoretically, they will have the same manifestations. But however, if there are additional changes in the neighborhood of those stats,
Right.
that could alter the impact. Actually, I did not go into details, but there are several modifier factors.
Right.
So that also, that impact is there.
Right. Right. So thank you so much, Professor Radha. And because we sort of wound up a little sooner than we anticipated. So if anyone has any questions, may I share your email ID with them if they want to write to you and want to understand something in greater detail?
Sure, no problem. I'll be happy to
And I will share
answer
your
to.
email ID if anybody wants more information about this. And if you allow me, I will thank you again for such a fantastic, intricate, and novel thing, a new area which you really held up before us. And then I will invite today's next speaker, who is Professor Elora Sen from NBRC, Manesar, Haryana. Professor Sen,
Gargi, Dr.
you're with us.
Gargi, if you can,
Hello, everyone.
Dr. Gargi, if you can give me few minutes or few seconds. I really enjoyed interacting with you all, and you have been so kind and inviting me, and I enjoyed, being here, and hope to meet you all in person sometime when I visit.
Professor Radhaji, we invite you to visit Amity University whenever you are here in India, and also our Long Island campus. When you go to the East Coast, we have in the New York Long Islands a beautiful complex which we have acquired, and so we want to develop that as Amity Global Research Hub. So please visit our Long Island's campus also. Thank you so much.
Sure, it will be my pleasure.
God bless
Thank you very much. Thank you. Bye-bye. Thank you, Dr. Gargi.
Thank you. Thank you so much, Professor Radha. Delight to have you among us. So I will request Professor Elora Sen now as a speaker. So I just want to introduce her formally to all of you. So in non-corona days, she is actually our next door neighbor, so she could have visited Amity easily, I guess. So she is from National Brain Research Center, which is in Manesar, Haryana. So Professor Sen, she received her PhD in immunology from Institute of Chemical Biology, Kolkata, and following which she went for postdoctoral research in the Penn State University, and where she studied transcriptional regulation of viral oncogenes. And she spent, I think, 5 years in Penn State. And after that, she did research associateship in neuroscience from Rutgers University before joining NBRC. Currently, she is scientist 6 and a professor at National Brain Research Center, and she has received so many awards. And if I had to name a few of them, the Kshanika Oration Award from ICMR in 2016, then Indian Society of Neuro-Oncology ISNO President's Award for Outstanding Work in Neuro-Oncology. She is also an elected fellow, of course, of National Academy of Science and an editorial board member of so many reputed journals. So I invite Professor Sen to give us a glimpse of the work that she has been doing for the last several years in her area of research, and she focuses on another very intricate, beautiful organ, the brain. So Professor Sen, if you could kindly share your slides and start the talk, please.
So, Professor
At the outset,
we are very proud to see such young dynamic leaders in science and technology coming and contributing to Indian science and technology. And we would like to connect with you. We have seen your outstanding contributions in the field of glioblastoma brain cancer research, and particularly with regard to the inflammation metabolism axis. You have focused on that. So we will be delighted to listen to you, and we would like to connect with you also. Subsequently, for collaborative research, we want to sign an MOU with the NBRC. So we have already moved a paper, and I think now your new director is in position, and we will take it forward. Thank you so much, and welcome to this webinar, Professor Elora Sen.
Thank you so
Celebrity.
Thank you.
Yes.
Loraji, welcome to METI, of course. And
Thank you.
Since we are neighbors, Professor Murthy, we get the priority of MOU with her first.
Great, Rajendra Prasadji, great, it's a great thing. And I know Ilora from a long time, but it was a chance that she didn't join in my institutes when I was director of ICPO. So that was because there was no post. So then she joined NVRC. It's a great that she joined there. And Ilora, nice to see you.
Thank you.
And her husband is also visitor to our institutes here in the AINN, Anil Pan and all. So I know them all. So nice to see you. And I know you have been doing excellent work, and it's nice to listen to you. Thank you. Go ahead.
So thank you for inviting me and having me here with you today. And as Gargi has mentioned, and Dr. Das has also mentioned, that I am a next door neighbor. So without going into much details, I will straight away go into what we have been doing over the last 15 years or so in my lab. And I'll try to share the screen. Yes, sure. Okay.
Perfect.
All right. So today I will be talking about three important components that characterizes the tumor milieu, microenvironment, the tumor milieu. One is the inflammation, and the other metabolism. And the third, I will be introducing you today. I'm sure most of you are aware of this, but I will show this in a different light, how a circadian rhythm kind of links between inflammation and metabolism in cancer. And being at the National Brain Research Center, our lab focuses on brain tumors. And the brain tumors, or the gliomas as they are widely referred to, they can be of the astrocytic origin, oligodendroglial tumors, oligoastrocytic tumors, and ependymal tumors. And but among this, glioblastoma multiforme are regarded as the most malignant of all the gliomas, of all the malignant brain tumors. Why so? Because they are not only widely invasive and infiltrative, they have this inherent tendency to recur, and they have a malignant progression. But importantly, most importantly, they are resistant to conventional form of therapy, that is temozolomide, which is the first line of treatment in glioma. Now, way back in 1947, Otto Warburg came upon this concept, which is known as the Warburg effect now, where it has been shown there is a dysregulated metabolism in cancer cells. Now, as opposed to a normal cell, you can very well imagine a cancer cell will need much more nutrient to sustain itself. Okay? So under normal condition, in the presence of oxygen, we find there is oxidative phosphorylation there is going on. But this is in case of a normal tissue, differentiated tissue. In the absence of oxygen, there is a conversion of glucose into the lactate. But what happens in the case of tumor cells, even in the presence of oxygen, glucose get converted into lactate. So tumor cells are characterized by the high presence of lactate, which contributes to its very high acidic pH in the tumor microenvironment. So first thing we have to remember that the metabolism is dysregulated in tumor cells, which is characterized by the Warburg effect, which is nothing but that even in the presence of oxygen, tumor cells produce lactate instead of undergoing oxidative phosphorylation. Now that was the dysregulated metabolism. Now we have something in tumor which is the aberrant inflammation, excess production of pro-inflammatory cytokine. And one of them, which has been a focus of our lab, is the presence of a pro-inflammatory cytokine, IL-1, interleukin-1. And way back in 2011, we have shown that in the GBM brains, in the GBM tumors, there is an elevated level of interleukin-1 as composed to the surrounding brain tissue. And IL-1 has been suggested as an important target for cancer immunotherapy. And studies from a group have shown that IL-1 can actually regulate the expression of hexokinase 2. And what is hexokinase 2? Is a metabolic enzyme of the glycolytic pathway through involved, and this regulation of hexokinase 2 by IL-1 involves a histone deacetylase like SIRTWIN6. So this is something that we have shown, that's a crosstalk between inflammation and regulation of metabolism. Also, a recent paper, we have shown that in the presence of inflammatory milieu, and in case of glucose deprivation, what we see is that there is a shuttling of hexokinase 2 into the nucleus. Now, as we are all aware that hexokinase 2 is a glycolytic gene. So what is it doing in the nucleus? Then what we found out, in the nucleus, hexokinase actually acts as a transcriptional co-activator. Now, what does it mean? There is another transcription factor called the NRF2, which plays a role in redox regulation. But what hexokinase does is, hexokinase binds with NRF2, and this complex goes and binds to the antioxidant response element on redox regulatory genes, and it regulates the expression of genes such as xanthine oxidoreductase. So what I'm trying to tell you is this: in an inflammatory milieu, there is a possibility that the metabolic genes can perform non-metabolic function as something that is coming in a big way, which is known as the moonlighting function of metabolic genes. So functions other than the metabolic genes. So here it is acting as a transcriptional co-activator. And this was the first time we were showing, yes, hexokinase does function in transcriptional regulation of gene. And this is beyond its ability to act as a metabolic gene. Now, I will take you to another component of the axis that we are going to talk today. That is the circadian rhythm. We are all aware what a circadian rhythm is, and whereby, you know, the day-night cycle, the feeding, fasting, that regulates a clock in the hypothalamus, which is known as the suprachiasmatic nucleus. Okay, so light dark circle, fasting, feeding, these regulates the suprachiasmatic nucleus and the peripheral clock. So in addition to the suprachiasmatic nucleus, which is a central clock, we also have other peripheral clocks in the body, in the adipose tissue, in the pancreas, liver, muscles, gut, kidneys. It's everywhere, the peripheral clock. Now there is even a crosstalk between the central clock and the peripheral clock, and this involves what? They involves hormones, humoral factors, glucocorticoid, the autonomic nervous system, the body temperature, insulin, free fatty acids, metabolism, you name it, and that connects the central clock with that of the peripheral clock. So what I'm trying to show, the organization of the circadian clock network in our body, regulated by the night-day cycle, as well as the feeding, fasting, appetite, and the metabolism. Now, there is a connection between the circadian rhythm and the cancer cell. There is an autonomous clock. Why am I talking about circadian rhythm in cancer? Because the circadian rhythm in tumor cells are totally disrupted. They have an aberrant circadian rhythm, And not only the suprachiasmatic nucleus or the pineal gland play an important role in the peripheral clock, but if you can see the arrows in green, you will find that inflammatory mediators like interleukin-6, such as IL-6, or the metabolites such as lactate play a role in maintaining this circadian rhythm. It's also involved. It's an important component of this whole day-night cycle, feeding, fasting cycle. So it's a part of the system. And so is also chemotherapy, like glucocorticoid. It can affect the circadian rhythm. So what I'm trying to bring here is that there is number one, there is a dysregulated metabolism, there is an aberrant inflammation, and together there is a dysfunctional circadian rhythm that is going on in tumor cells. Now, in addition to the peripheral clocks, each cell of a body have molecular clocks. Now, these molecular clocks are composed of several transcription factors and histone acetyl transferase, and I will just mention two of them. One is known as the clock and the other is BMAL1. Okay, now why do you think circadian rhythm would be of interest? If you look closely at this, you will find that several of the factors that are associated with the circadian clock are associated with the cell cycle progression of the cell progression. Therefore, any defect, and it's because it's a coupling between the circadian clock and the cell cycle, maintains a homeostasis as to the proliferation of the cell, a cellular homeostasis. You can very well imagine that if there is a disruption in the circadian rhythm, you are bound to get an altered cell cycle. So a disrupted circadian rhythm affects the cell cycle progression. Now, a paper have shown, recent paper in Cell paper, which have shown that the pH of the tumor microenvironment plays a very important role in the circadian rhythm. Now, so we have two things. We have the central clock, suprachiasmatic nucleus. We have the peripheral clock in all the tissue, and again we have the molecular clock in each cells of the body. And the pH of a cellular microenvironment affects the circadian rhythm. Now, because tumor cells have elevated levels of lactate, and lactic acid contributes to heightened pH. So the next obvious question to us, is there a link between this altered pH in cancer cell, altered circadian rhythm and inflammation? Because we have already shown that lactate can regulate IL-1 can regulate the metabolism. So given that tumor lactate dehydrogenases play an important role in tumorigenesis, the central role of lactate in glucose deprivation is coming up in a big way. And we have shown that IL-1 regulates metabolism. So what we asked is what the question next we asked, where does circadian rhythm fits into all this? Because we know what are the emerging hallmarks of tumor cells. Besides the genomic instability or immune evasion, we have the dysregulated cellular energetics, that is nothing but altered metabolism, and we have inflammation here. So the next question we were asking, where does this circadian rhythm fit into the whole scheme of things? So this is a question. Does the tumor promoting environment, inflammation, dysregulated energetics, and desynchronized circadian rhythm, is there a crosstalk going among all of this? And what is the role of the clock and BMAL1, which are the main component of the molecular clock? And what is the role in regulating the metabolism, inflammation, circadian circuitry? Now, to address this, we chose a very simple tissue culture model whereby where serum shock induces circadian gene expression. So it is a very simple system. We took the cells, we exposed them to serum deprivation, and that is a model of a synchronization and synchronizing the circadian rhythm. So now with that, what we did was we treated the glioma cells with sodium oxamate because glioma cells already have elevated level of lactate because of the Warburg effect. Now we wanted to see what happens when you inhibit the lactate, and sodium oxamate is a lactate inhibitor. You can ignore the Western blot because here it's getting too crowded, but if you look at this densitometric representation of this Western blot, you will figure out that inhibition of lactate affects the rhythmic expression of clock, BMAL1, and interleukin 1. So when you inhibit lactate, there is an inhibition, there is an altered regulation of the rhythmic expression of clock, BMAL1, IL1, and of course lactate dehydrogenase. Okay. Now, since inhibition of lactate was affecting the release of IL1, so the next obvious question was, if we treat, so we were seeing this lactate inhibition is regulating IL1 and the clock. The next obvious question was, if we treat with IL1, what would happen? And we found the clock, BMAL1, lactate dehydrogen follows a rhythmic, cyclic rhythmicity when you treat the cells with IL1. So again, what we show here, that just like lactate was regulating IL1 and clock, now IL1 is also regulating lactate and the molecular clocks in this cell. Now we did a couple of knockdown of clock and BMAL1. Once again, what is clock and BMAL1? Clock is a histone acetyltransferase. BMAL1 is a transcription factor, is a basic helix loop helix transcription factor. And this complex of both clock and BMAL1 as a heterodimer, it will come and bind to the EBOX sites of clock regulated genes. So clock genes has an EBOX site. Now what we did was we knocked down clock, we knocked down BMAL1, and what we find, there is a decrease in lactate dehydrogenase, there is a decrease in IL1 in both the cases. Now, when you derive a BMAL1 mutant, which lacks the basic helix loop helix, whereby it's no longer able to go and bind to the EBOX, also you find a decrease in lactate dehydrogenase, there's a decrease in the IL1 expression. So we were trying to say is IL1 and lactate behaves as a clock BMAL1 target. Okay. So they are acting as targets, lactate and IL1. So we have shown this by lactate release assay and how the different combination is affecting the release of lactate. For example, IL-1 causes a release in lactate, and when you treat with IL-1 inhibitor AF1298, so there is a decrease. Similarly, when you treat with clock siRNA, BMAL siRNA, or lactate dehydrogenase siRNA, the increase in the lactate levels, it goes down. So what we're trying to point out here is very simple, that lactate and IL-1 are clock BMAL1 targets. So these are regulated. So one is a pro-inflammatory cytokine, the other is a metabolic gene. They are regulated by clock BMAL1 targets. Now, to go delve a little deeper. Now, this clock and BMAL1, they form a heterodimer. And what happens then, it goes, the BMAL1 being a basic helix loop helix, goes and binds to the EBOX sites on the genes that are associated, regulated by clock. So clock genes, usually clock regulated genes have this EBOX sites where the clock BMAL1 can come and bind. Now, clock has a histone acetyltransferase activity. Now, when clock acetylates the histone marks here, increased acetylation is accompanied by diminished methylation, resulting in the opening up of the sites on the EBOX and allowing the clock BMAL1 complex to come and bind here. Interestingly, what we find it was very excitement when we first found that yes, both the IL-1 promoter and the lactate dehydrogenase promoter does have EBOX sites on their gene. So that is kind of an indirect proof that yes, IL-1 and a BMAL1, IL-1 and lactate can be BMAL1 clock target, but we had got to prove it. So what we did point mutations across all these EBOX sites, and we found that mutation of this EBOX sites reduces the IL-1 or the LDH promoter activity upon treatment with lactate or IL-1. So basically, this EBOX sites on both IL-1 and the LDH promoter play a very important role in the regulation of these two very important genes of the tumor microenvironment by clock and BMAL1. Now we did another very simple experiment. We just did a chip and followed by a rechip. Now first BMAL has to bind to the EBOX sites, followed by the clock. So we did a chromatin immunoprecipitation and followed by a rechip by clock. And we found out, yes, on this EBOX side, clock and BMAL1 is actually binding both on IL-1 and LDH promoter. I'm not going into greater details, whereby we have shown how change in the histone methylation or the acetylation affects this binding of the clock BMAL1 to the EBOX. Once again, clock is a histone acetyl transferase. And this heterodimer is necessary for exhibiting the functions of molecular clock. Now, so if there is a clock BMAL1 circuitry, which is cross-talking with your lactate and your inflammation, so what does it have to go to do with tumor progression? So at this time, students who had a great deal of knowledge on bioinformatics, what she did, she found this paper in glioma where they have done a ChIP-seq and an RNA-seq of 9 GBM patients. And what we did that was, we tried to find, because we were finding that clock, BMAL1, histone methylation, histone acetylation, and histone methyl transferase was involved in the LIC circuit. So from this ChIP-seq and RNA-seq data, we tried to find out the intersection between all the 3 components that are involved in the LIC circuit. And what did we find from that analysis of the data? That histone modification, cytoskeletal organization, DNA repair, chromatin organization, DNA damage, cell cycle, every event that is crucial for gliomagenesis is actually affected by this clock, BMAL1, and the methylation acetylation circuitry. So then what we did, it was a very simple experiment where we just looked into treatment with sodium lactate or IL-1 or sodium oxamate to inhibit lactate, treatment with clock siRNA, BMAL siRNA, LDH siRNA, and IL-1 in the presence of IL-1 antagonist. And we looked for all the markers that I've shown you in the previous slide, like the cell cycle, DNA repair, DNA damage. And what we find is truly that this clock BMAL1 regulates the molecules that are associated with events that are associated with tumor progression. So this is a take home. To very simplify this, what we are showing, that binding of clock and BMAL1 on the EBOX involves a lot of chromatin remodeling going on this EBOX side, and it results in the expression of lactate dehydrogenase, release of lactate, and this lactate dehydrogenase, again through the release of lactate, again it facilitates the release of IL-1 against lactate. So we are talking about a cycle. There's a feed-forward loop that is continuously going on to maintain a certain level of IL-1 and lactate dehydrogenase that ensures that the cell cycle, DNA damage, cytoskeletal organization, chromatin organization, everything is maintained at a level which is sufficient to maintain the glioma state in a state of proliferation. So these are the events. Now, so what? So what if we named this circuit as the LIC circuit, like a life insurance corporation. We got the term from that, and we like LIC is the lactate, IL-1, and the clock. And what we find when we reanalyze the TCGA data set for patient survival, what we find out that it has a direct correlation with the patient survival. So more clock, more BMAL1, more LDH, and more IL-1 in GBM patient indicates a poor prognosis. Now, if this is true in glioma, does it hold true? Is the LIC circuit present in other cancers? Yes, to a surprise, we found that this circuit, it's present in almost all the solid cancer, but we found just a reverse correlation in hematological malignancies. But the expression, the correlation between IL-1 and LDH was present across several tumors. And I'm not sharing the data, but what I'm showing is that that this LIC circuit, the lactate, IL-1 and clock circuit is present across cancers, except hematological malignancies in all solid tumors. And what does that mean now? Now that we have come with the LIC network, then we taking these these components of the LIC network, that is clock BMAL1, which is an heterodimer, operates in a heterodimeric form. You have IL-1, you have LDH. Now we use something called the analyzed data from the genomics of drug sensitivity in cancer database, which is a GDSC data. And we found that this LIC network actually correlates with several other signaling pathways, which are actually the targets of which are very good anti-cancer targets, like EGFR target. Okay, you see in red means that this EGFR target has a negative correlation with the LIC circuit. Then your this EGFR signaling, when it is regulated by gefitinib, it has a positive correlation with this circuit. But your P53 has a negative correlation with the LIC circuit. Therefore, if a patient has been treated with gefitinib, okay, it will give a positive response, because when you inhibit the EGFR signaling using the gefitinib, it will inhibit the LIC circuit. But if you come ahead and treat with a P53 inhibitor, if you are inhibiting the P53, actually you are inducing the LIC circuitry. So from this correlation, we have come upon how this LIC circuitry can be correlated with pathways, and those pathways that are again correlated with drugs that are already approved in the clinic or in the stage of experimental trial. So for our case, we have pinned down with gefitinib, because in glioma, EGFR signaling is amplified. It is mutations is very common, and if you inhibit gefitinib, you not only inhibit the EGFR signaling, but you also affect the LIC circuit. So this was we were trying to correlate the LIC circuit with your drug sensitivity. So therefore, the genomic landscape and the pharmacogenomics of clogged genes is now a widely discussed topic in cancer chronotherapy. And now that we have this LIC circuit showing a correlation between LDH, IL-1, clock circuitry, we are trying to fit it into a mathematical model in collaboration with Mithun Mitra from Department of Physics in IIT Bombay. And we are trying to predict exact time points whereby, targeting, say, a particular rhythm could bring down the lactate levels or the IL-1 levels. So as I just mentioned, what is chronotherapy? So treatment at certain times during a circadian rhythm will have the optimal chance to optimal effects in showing its manifestation of anti-tumor targets. For example, patient A, B, C. So there are many different circadian rhythm, as many as a patient. So the cancer chronotherapy is coming up in a big way, where they want to target a particular phase of the circadian rhythm, where you treat with different inhibitors, or you treat with the LIC components of the LIC circuit, or the signaling pathways that are affecting the LIC circuit to achieve the optimal effects of your anti-tumor drugs. So in a broader sense, our LIC circuit does have a greater implication in drug administration timings, and we are kind of trying to work out the mathematical modeling, and hope I have been able to convince you that such a circuit does exist in tumor, and it plays a very important role. How now we are showing a crosstalk between your metabolism. I'm showing a crosstalk between metabolism, inflammation, and desynchronized circadian rhythm in the tumor cell. So this work was supported as a unit of excellence in cancer biology by Department of Biotechnology. And I will stop here for this part of the talk. And if anybody has any questions, I can take it now, or if you want, I can move on to the next section, which I promise is going to be even more interesting. Should I go ahead, or should I?
Professor Ellora, I think.
Now, Ellora, could I have one questions now?
Yes, please.
Yes, it's an excellent thing which I have been long time looking, and I have some other contributions in this line, but it interests me a lot. But I happens to visit France in Paris long back, the Institutes of Chronopharmacology. So and then I always talk of here many of the occasions that the treatment to the doctors, being in ICMR earlier, that the treatment of the medicine, so that should be on the basis of what is now your slide is showing, the times during the circadian rhythms to be taken into consideration. Even we know that even the homeopathic medicine which is given just in the morning and the so that has some relation to the circadian rhythms. So that affects much. And what I learned earlier, that the drug treatment has to have time when the cell cycle has to taken into consideration. I didn't see anything considerations with the cell cycle.
So we have shown it was a very busy slide, probably.
Okay, I missed that. So, and it's very important, but your title is very interesting, that it is the inflammation, and your clogged gene, that the BMAL1, so genes that is associated with the tumor progression. But is that the BMAL1 is the anything to do with the NF-kappa B, the pro-inflammatory? It is the downstream or upstream, or it is interacting, or if there is a crosstalk between the.
A recent study have shown that BMAL1 clog doesn't really have anything to do with NF-kappa B signaling. Very recent.
No, but then you have the clogged genes have the relations with the inflammation you are telling.
Yes. So
inflammation in cancer is very interconnected.
No. See, BMAL clock 1 might not directly affect NF-kappa B, but by regulating IL-1, it can induce NF-kappa B. So it does in an indirect sort of way.
Yeah, because that's what I was wondering. It may not be this gene, but it should have some indirect connections with the through other IL and other things. So because of the inflammation and cancer have the basics of the genetics. Okay, I think.
Hi, Ellora. This is a journey.
Hello.
Hi, Ellora.
Yes.
Good, nice talk, nice talk. So I have a quick question that when you talk about the central clock, the peripheral clock, and the molecular clock, and the synchrony between them, where does intermittent fasting fit here? Does it fit? Does it help?
Yes, intermittent fasting does fit, because when you're talking about the glucose level does fit in. Intermittent fasting, that's fit in, because you are depriving yourself with glucose metabolism, and then you get an upsurge of glucose, following your termination of fasting. So yes, intermittent fasting does fit in.
Because intermittent fasting would affect both the inflammation as well as the tumor progression.
Sure.
So a mouse model of that would be really nice.
No, see, we thought of it, but you see, getting a mouse model, getting it into a circadian rhythm, injecting it tumor, and then following, it's not possible. We should acknowledge the fact that sitting here, it is not really possible in this country. Maybe, and it involves several groups who can bring about such a, coordinated approach is required, not a single group by myself.
Yeah, because I had seen a couple of studies by Walter Logo. So maybe, I mean, it's just that, it does fit in very nicely. That's why.
Yeah, it does. And, it's under revision. The paper is under revision. And actually, they really appreciated that, the clock actually goes and binds to IL-1 and LDH, which was we are showing for the first time.
Thank you.
Madam, good afternoon. Chandramani Pathak here.
Hi, good afternoon.
Ma'am, just I'm actually interested about is this a signaling is stated with activation of the inflammasome for activation of interleukin-1 beta?
We have not got into the inflammation, because we are mostly interested into the gene regulation and chromatin remodeling, which I have not shown in greater details. We were into the nucleosomal opening up, then acetylation. So we really didn't want into the inflammasome component. No, we didn't.
Okay. Thank you, ma'am.
So maybe can I move into the next?
Yes, please.
Yes, okay.
Yeah, please continue.
Yes. So now I had requested Dr. Ujjaini Dasgupta that I will be talking about this, but we had a very interesting finding from our lab, which I must show, because if not for anything, but for the sake of the scientific endeavor that went into coming to the conclusion. So it is related to COVID-19, and during the first phase in April, May, we were asked by DBT. It was a part of the DBT initiative that NBRC, think of something to contribute to the nationwide program on COVID. And at that time, given our, the basic theme of the lab, given that is inflammation. So I thought, why not focus on the inflammatory aspect of the host response rather than the viral component. And as you are all aware that this COVID is associated with uncontrolled inflammatory responses, and these are the clinical features ranging from fever to acute phase protein, acute kidney injury, elevated serum, decreased serum protein, and and every other day somebody is coming up with a newly previously unknown characteristics of this disease. So this exaggerated host inflammatory response, which results in a cytokine storm, is a major cause of lung injury and subsequent mortality in severe condition of COVID-19. So with that theme in mind, so we set out to dissect out a few points. So there is an elevated cytokine storm which is known, and there is something called the macrophage activation syndrome, where the macrophages becomes hyperactivated, and hyperinflammation, as I already mentioned, severity of the disease manifestation. So the obvious choice is, are there any anti-inflammatory in the market which could be a viable strategy? Now, first we did very simple experiment. We took took a BS2B cell line, which is human lung cells, and we treated with them with the viral proteins. And I will mention only the 3 viral proteins we have here. That is the spike protein, the envelope protein, and ORF3. So what we find that when these cells are exposed to, we overexpress these viral proteins, we find there is an induction of cell death, lung cell death. And there are reports of there's a cytokine storm. So in these cells which are undergoing cell death, we measured the release of cytokines from the cell, and we found there's an elevated expression of interleukin-1, again, you know, like inflammation here, that is a high levels of pro-inflammatory IL-1 that is released, together with a very important pro-inflammatory cytokine called high mobility group histone protein. As the name suggests, is a histone. It modulates, you know, chromatin structure, is a histone modifier. And we have been working on this lab, but from a different angle. But what is known that HMG1 potentiates the activity of IL-1. So now there is an elevated expression of histone, is extracellular release of histone HMGB1 together with the release of pro-inflammatory cytokine 1. Now, release of cytokine 1 has been regarded as an important mechanism to drive pyroptosis or inflammation mediated death. And now we question whether this increased cytokine is accompanied by pyroptosis, and we found there is an increase in caspase-1 activity, and this caspase-1 activity is necessary for this cleavage of this pro-inflammatory into this mature form of IL-1. So what we are showing that the viral protein induces pyroptosis in lung cells, and it is accompanied by the release of HMGB1. Okay. Now, HMGB1, there was a release of HMG1 more in the case of when it are transfecting with ORF3A. Now, HMG1 inhibitor glycerizing is widely used by people working in histone modifiers to regulate the effect of histone HMG1, and we found that when you treat with glycerizing, it prevents viral protein induced pyroptosis. So there is pyroptosis when you treat with glycerizing. There it rescues the amount of death. There is also a rescue in the release of the caspase, and when you treat with glycerizing, there is a decrease in the HMG1 release, as you can see here. Now, any infection is bound to attract, the immune cells to a site like monocytes, macrophages. And what we wanted to see, if you take the supernatant from the BS2B cell, which are overexpressing viral protein, if you put them on the monocytes, whether differentiated or undifferentiated, what is the status of the cytokine? And we found that, yes, there is further increase in HMGB1 from the monocytes or macrophages when you treat them with the supernatant that is released from the lung cells expressing the viral protein. So once again, so when you take the supernatant from the lung cells overexpressing the viral protein, you get a high expression of HMGB1, IL-1 cytokine, and when you treat with HMGB1 inhibitor, there is a decrease in the expression of HMG1 and the other cytokines. So glycerizing and HMGB1 inhibitor reduces viral protein-induced macrophage activation. Around this time when we were doing the experiments, findings suggested that in some patients, there is an elevated release of ferritin. And ferritin plays a very important role in inflammation. So the next thing that we did was we looked into the ferritin level, and there is an enormous surge of ferritin release from the monocytes, which are exposed to this supernatants that from the lung cell. And this ferritin surge was rescued in the presence of glycerizing. Now, so I once again, I emphasize 3 points. One is that viral protein induces pyroptosis of the cell, lung cell death. It causes a release of cytokine. It causes a release of cytokine. It causes a cytokine storm from monocytes, and it is inhibited. The cytokine storm pyroptosis is inhibited by glycerizing and HMGP1 inhibitor. Now, the next step is, does glycerizing have any effect on the viral replication? Because we are talking about viral proteins till now. Now, the next what we did at Regional Center for Biotechnology, they have this anti-COVID screening facility. So we sent glycerizing, and they came up with a wonderful result where shows that when you infect the VeroE6 cells with these the SARS-CoV-2 clinical isolate, and we find that glycerizing, even at this highest dose, had no effect on, no toxicity on Vero cells, but it was completely able to prevent viral replication, and its efficacy is as comparable as remdesivir. So in addition to its anti-inflammatory effect that we have seen, it also have an antiviral effect, and without inducing any toxicity to the Vero cells in which the live viruses were there. So what I want to emphasize, that glycerizing have an antiviral effect as well as an anti-inflammatory effect. So this was published last month in the Cytokine Journal, which is a official journal of the International Cytokine and Interferon Society, where we find that when you inhibit HMGP1 by glycerizing, you not only prevent HMG1 release, cytokine storm, but you are 100% capable, or not 100, about 90% capable of inhibiting the viral replication. So this finding was reported by Nature India, and it was at this point when we were communicating to Nature India, it dawned on us that glycerizing is an active component of licorice root or yashtimadu or muleti, which is widely used in Ayurveda to treat respiratory problems and inflammation. So I want to highlight two important aspects here. We started off very innocently as an HMG1 inhibitor, and we nailed it down to yashtimadu or muleti, which is an HMG1 inhibitor. But survey of literature, what we found that in 2003, when there was a first SARS associated virus outbreak, glycerizing was the most active in inhibiting replication of SARS associated virus. So when we were about to write the paper, we figured out that it was already shown in SARS associated virus. When we had communicated the paper, the same month, this paper came out in Cell, where they find that HMGB1 is a novel regulator of the AC2 expression or a viral entry. So what we targeted as an inflammatory inhibitor of inflammation, inhibitor of HMG1, and it was very, very heartening to and heartwarming to find that others have corroborated our finding through different angles, and HMGB1 is actually a novel regulator of AC2, and we find that this herbal compound not only stops inflammation and viral replication. And I thought that I would do injustice to our Indian traditional medicine if I do not discuss this work, because what started off as an inhibitor of HMG1, which is actually a herb that is used in Ayurveda, might contain a drug against COVID-19. And this was reported by Deccan Herald. And I tried my best so that it reaches as as many people as possible. So in Maharashtra and, in the Maratha, this is a Shakal newspaper, in Dainik Bhaskar, and all, and that is in Anandabajar, a very popular newspaper in Bengal. So this is my only way I can, make people aware that this may be considered as a has potential. I'll stop it at it as a potential. And because in the northern part of the country, the people do chew on Yeshtima muleti in the winter months, especially for dry coughs and for sore throat. So I wouldn't recommend that everybody starts chewing it, but I'm just putting forth my view that it does have potential, because it's not only us, but the HMG1 ACE2 has been shown by other group as well. So with that, I end the corona part, and what I started of an inflammation showing in the context of the tumor. I have also nailed down inflammation in the context of the COVID infection. But what I would really want to say that nothing in this universe is unconnected. So there is a great deal of dependence on one system on the other, and where the boundary actually merges, there is actually no boundary, and it's a mental concept. There is a boundary between your inflammation, your metabolism, your epigenetic, your genomic landscape. What we are actually showing that this universe is really an inseparable web where interconnections are dynamic, and it's I've taken it from the Tao of physics, and very much like our ELISA circuit, which is very much there, exists, and is very much alive. So is a cosmic web, the interdependence among inseparable, the wholeness of one. It's everything is beating in the same rhythm, I would say. And so we are actually all connected. With that, I would have a really small lab, but we are very interconnected lab. And this is our lab pet. His name is Chom Chom from and with that, I would like to take in any questions.
So, Professor Elora, thank you so much for both the aspects that you highlighted today. And firstly, you talked about inflammation and the circadian clock and cancer, and which is certainly a new dimension and a new way of understanding that. And I invite the panelists to ask any questions regarding that. And of course, the second talk is equally thought-provoking, and I personally am really excited that you highlighted an Ayurvedic component, and you actually, you said that others have also done it, but your team was able to bring it to the forefront of research, and I'm really very glad to see that. So I invite all the panelists to ask Professor Elora questions that they might have.
Yeah, I will ask just one more question. Is hexokinase 2 can modulate the Knox family of protein for reactive oxygen speed generation?
Yes, it does. We have shown that in the JBC paper.
Elora, one more question. Did you ever check the role of ceramide in your crosstalk between this desynchronized clock and inflammation? Because ceramide.
No, we haven't checked on ceramides, but a couple of years back, we had looked into the role of fatty acid metabolism and lipids in regulating the inflammatory network and DNA damage responses. And we had access to patient samples whereby we showed how lipid metabolism regulates, is affected by the epigenetic landscape. No, we have not looked into ceramides.
Okay.
Elora, I just had one question, maybe a bit naive, but still I would like to know. Of course, you have shown and others have shown that lactate is a very important component of the tumors, and the BMAL and the clock, which you have shown, they bind to those two genes. My question is, although you did show that they regulate so many networks, but so do you see a substantial change from normal people versus patients? You did show the analysis, I know, but I am just wondering how different is it in the patients versus the normal people? Because if these two genes are regulatory and they bind to the E box, right, as you mentioned.
Yeah, I have shown the TCJ data set, right? If probably you have missed it, where we have shown that clock and BMAL gene are altered in patient as compared to the, they are totally altered.
Totally altered, all right, thank you. I might have missed the slide. Thank you. Ujjani, if you would like to then thank our speakers and take over to thank everyone.
Yeah, sure. So we had a nice morning. It started with a little bit of technical glitch, but I don't think that really mattered, because the essence of it was good science, and not only in the perspective of cancer, but also the very recent pandemic situation. And with that, I would like to conclude the session by thanking everybody. And I thank Professor Selvamurthy, Professor B.C. Das, and our director, Professor Rajendra Prasad, for being with us, for encouraging us and actively participating. I also thank the speakers, Professor Radha and Professor Elora, for giving us two completely different perspectives. We learned about the very rare mutations that bring about retinal degeneration and the molecular basis of it, which is a holistic concept, because rare mutations are something that happens 1 in a million, but it happens, and it is very difficult to nail them. So that was the first part, and the second part was by Professor Elora Sen, and she beautifully explained the crosstalk. She talked about the peripheral clock, the molecular clock, the cellular circadian rhythm, and then beautifully amalgamated them with inflammation and tumor progression. And the best part of it was it actually not only is restricted to glioblastoma, but it is restricted to, it is not restricted, but it is also in different types of other solid cancers, which makes one understand the depth of the depth of the mechanism and the depth of the understanding of these crosstalks. And second thing was that she talked about the role of glycorizin in inhibiting viral replication and inflammation. And so the take-home message would be to chew on mulethi, which I think doesn't hurt us anyways. It is not expensive. It is not harmful to the body, and it's just I mean, it can just be taken up as a little condiment. So with that, I would like to thank all the panelists, my colleagues, my colleague, Dr. Gargi, for being there together. We tried to arrange, and the other panelists, my colleagues, Dr. Amit, Dr. Munindra, Dr. Pathak, and definitely the IT team. Our Amity Noida campus people, our Amity Haryana, and the rest of the Amity family. And with that, I would say that thank you for being here, all participants. And let's hope we have another session soon. Let's hope someday we all meet in person and we discuss some very good science. And with that, I would conclude the session. I would thank everybody and stay safe. Thank you.
Thank you.
Thank you.